In this work, we present an atmospheric model tailored for the interactive visualization of planetary surfaces. Our model provides a complete framework for the absorption of light by small and large particles, such as the oxygen and ozone molecules and Mars dust particles. We demonstrate our results using Earth and Mars as examples.
Nowadays, technology is developed for a single audience, assuming that one size fits all. Nevertheless, we all have individual differences, such as personality. Taking into account how personality affects preferences, we study whether it has an effect on hierarchy, evolution over time, and comparison contexts regarding information visualization. We leverage all personality variables from the 5-factor model and the locus of control with correlation and clustering approaches, showing promising results.
This study presents a method to explore hierarchical data by improving ROD-VIS, one of the visualization techniques for multidimensional data. The user can set options for nodes and check the data in groups or individually in the ROD-VIS. 3 subviews at the right enable the hierarchical clustering structures of selected data and allow the user to grasp the features of selected data. As a result, this system allows more detailed insight into the data while solving the node overlapping problem.
Existing visualization tools for deep learning classification are mostly working on natural images with mutually exclusive classes. The classification of X-ray scattering images is more challenging since their multiple structural attributes have complex relationships. So, we built an effective visualization system to study the model performance and to find questionable labels or outlier images for scientists' further improvement.
Designed in response to the COVID-19 pandemic, the novel visualization technique we are calling GOWNS accommodates continuously incoming and increasing case data without distorting past data. Layering linear scale graphs shows both small and large numbers. This allows more detail to be seen when scrubbing through time and comparing regions by realigning the GOWNS based on case numbers.
Data GIFs are enjoying increasing popularity on social media as a format for data-driven storytelling with visualization. Simple visual messages are embedded in the short animations and are played in automatic repetition. Here we present a structured design space and a set of design instructions to create effective data GIFs.
We propose V2V, a comprehensive pipeline for variable selection and translation for multivariate time-varying data. Our approach contains feature learning, translation graph construction, and variable selection. To demonstrate the effectiveness of our approach, we compare histogram matching, pixel to pixel, and cycloGAN to evaluate variable translation. In addition, we also compare an information-aware framework to evaluate variable selection.
What is insight? It is a question that has bothered visualization researchers for a long time. We're wondering whether visualization practitioners think about data insights in the same way as visualization researchers do. So we interviewed 23 visualization practitioners to understand how they think. To find out more about what we found and the implications to the visualization community, take a look at our paper.
Hi, and welcome everyone to the session on molecules, cells, and vessels. My name is Michael Krone, and I will be chairing the session. Here we will have 5 interesting talks, and the first one is HyperLabels, browsing of dense and hierarchical molecular 3D models. The talk will be given by David Kouřil, and this is a joint work between the TU Vienna, Miriah, the Masaryk University, and Coast.
Hi, my name is David Kouřil, and I will present our work accepted to DBCG this February. This paper was a collaboration between the visualization group at TU Wien in Vienna, Austria, and several other labs worldwide, as you can see here. In this paper, we present a method for browsing of 3D models coming from structural biology. And I'll right away show you what that means. We introduce a method for traversing complex virtual environments that are built up by multi-scale molecular models. Textual labels are often required for interfacing with users to provide annotation. Without these annotations, users might have no idea what they are looking at. We leverage the textual labels and turn them into active clickable elements, HyperLabels. We then allow users to navigate the complex hierarchical 3D structure by clicking on HyperLabels annotating individual components of the model. It's important to point out that our work is mostly aimed at novice users, those with little to no previous domain knowledge, such as members of general audience or beginning students. We therefore provide the ability to explore large molecular models in an intuitive way. Our solution is motivated by the characteristics of multi-scale molecular models, which can contain interesting features on several levels. In the simplest case, however, when we render the model as a whole, we do not get the correct impression of the internal complexity. The outer layer occludes all the internal objects. There are ways to show these internals, but they mostly decouple the exploration of the model's hierarchical and spatial aspects. We identified 3 categories of related work from the area of visualization and computer graphics. First, labels have been extensively used in visualization, and in some cases also the clicking interaction was included. None of the existing work, however, uses the clicking interaction for traversing a hierarchical organization of a spatial model. The second category is navigation and exploration in 3D multiscale environments. These works mostly feature relatively sparse models and do not dynamically adjust visibility of the individual scene elements to fight occlusion. The final category of related work therefore deals with occlusion management. In the paper, we review techniques for polygonal, volumetric, and molecular data. Based on our research, we believe that the way our method tightly integrates the dynamic resolution of visibility with the navigation is uniquely novel. We therefore propose a technique that enables to browse the 3D model, similarly to how one browses a web page or a file system. The examples of web and file system browsers serve as an inspiration to our technique, and we use certain design elements from those environments. We leverage the fact that such concepts are well understood by most of the general public. Altogether, our method for browsing of 3D molecular models consists of 3 components. First, the previously mentioned hyperlabels are the central element and serve for a top-down navigation, diving deeper into the hierarchy of the model. Second, a breadcrumbs panel, which on the other hand enables bottom-up navigation, essentially stepping backwards. And third, a structure of interest opening procedure, in which the visualization is changed in order to showcase the selected structure. The structure of interest opening is the key process that enables further recursive exploration of the molecular model. Next, I will talk a bit more about these 3 components. Hyperlabels serve several purposes. First, they fulfill the annotational role of textual labels in that they connect shown structures with their annotations. Furthermore, they are interactive, that is, clicking on a hyperlabel selects the associated spatial substructure of the model. The name suggests our inspiration for this concept, hyperlinks from the web. Similarly to how a hyperlink transports the user to a new location that is a new web page, a hyperlabel too takes the user to a different location, in this case a different component of the large and complex model. The primary purpose of the hyperlabel interaction is selection of the current structure in focus, which we call the structure of interest. While hyperlabels enable a top-down navigation, traversing the hierarchy from the root to the leaves, breadcrumbs panel enables the opposite, to go up the hierarchy. Here again we are inspired by a UI element from web design. The breadcrumbs panel is a reduced version of a tree, showing only the path from root to the current structure of interest. This panel combines the purpose of providing orientation to the user, which is very much needed in a hierarchical multiscale environment that is on top of that 3D, and the interaction, which allows navigating to any structure from the breadcrumbs panel. Structure of interest opening consists of 3 procedures. First, we adjust the visibility of the model to showcase the selected structure. We are making the dense model on purpose more sparse. That's why we call this step sparsification. Next, we must change the camera viewpoint so that the selected structure is shown in the most appropriate zoom level. We therefore anchor the camera onto the selected object. Finally, we re-annotate the model with hyperlabels describing the current level. All these 3 procedures must be performed together so that the model is prepared for further exploration. The goal of the first step is to reduce the density of the model to showcase the selected subpart. More specifically, we open up the structure and show its internal components. To do so, we use the hierarchical organization. Then we take the advantage of the multi-instance character of the data. That is, the fact that every structural type is represented in a model by many instances. We perform the sparsification in 2 steps. First, we select a representative instance for each of the child structural types. Several strategies can be considered here. In our case, we use the heuristic of taking the instance that is closest to the camera, which worked reasonably well. In the second step, we resolve the final visibility. One option is to only show the single representative instances from the previous step. That way, however, we lose the contextual spatial information. For that reason, we also implement a strategy we call the context cut. With this approach, we place a cutting plane at the position of the child representative instance that is farthest from the camera. That way, we ensure that all representatives are visible, but also include some spatial context. Finally, the instances that are removed or sparsified do not necessarily have to be completely hidden. Here we show examples of various visual styles possible, such as ghosting or contour rendering of the cutaway parts. After sparsification, we highly likely end up with a scene that is a subset of what was shown before. In order to focus in on this subpart and show it in an appropriate detail, we adjust the camera. We position it closer to the selected subpart and embed it into its local coordinate system. This allows the user to use conventional navigation operations such as zooming or orbiting with reference to the center of the focused-in part of the model. To estimate the shape of the target structure, we compute a bounding sphere on the fly. We then use the parameters of the projection to calculate an optimal camera position to fit the bounding sphere maximally in a screen. As a final step, we change the annotation. A click on a hyperlabel conceptually represents a step in the hierarchy. In the re-annotation step, we therefore deploy hyperlabels that annotate structures on the new level. We then also update the breadcrumbs panel so that the linkage between the 3D view and the breadcrumbs panel is maintained. The breadcrumbs panel should serve kind of like a URL, specifying the location of what the user sees in the main 3D view. After the re-annotation step, the visualization is ready for further exploration. The user can either step further down the hierarchy by clicking on the newly shown hyperlabels, or go back by interacting with the breadcrumbs panel. We showcase the method on 2 mesoscale models from structural biology. These models uniquely represent the specific characteristics of being multi-scale, multi-instance, hierarchical, and very dense. We believe that the interaction method described in our paper can be translated to other models from other domains. The overall framework of hyperlabels will still be applicable. The only modification might be the specific implementation of the 3 steps of the structure of interest opening. For example, the sparsification method would need to be adapted for the specific model from a different domain. Naturally, there are a few limitations which we describe in the paper. Here, let me just shortly talk about one of those. The method we describe works on a step-by-step basis, and therefore doesn't allow rapid jumps between different parts of the model hierarchy. This was, however, a conscious design decision. We prioritized the navigation pattern used for exploration rather than finding specific structures and navigating towards them. We think that by supporting the stepwise interaction, we better help the user to make sense of the hierarchical organization as well as the spatial relationships between the components. To conclude and summarize, I presented our paper which proposes a method for browsing of a 3D molecular model using active textual labels, which we call hyperlabels. We integrate methods for navigating both hierarchical structures and 3D spatial structures and combine them into a technique that addresses annotation, exploration, and navigation at the same time. We find this way of navigation suitable for massively multiscale environments, such as biological models created to study life processes holistically on multiple levels, rather than researching them separately. In this work, we on purpose left out questions about efficiency and usability of our method. We believe this matter elicits a much more detailed follow-up work and should be the topic of separate future research. What would also help evaluate usefulness and intuitiveness of our technique is a deployment in a public space, which we would love to do in the future. Here are all the co-authors. On top of that, I would like to mention that this work was motivated by our long-term collaboration with scientists from Art Olson's lab at Scripps Research in San Diego, who served as consultants from the biology domain in this project. And with that, this will be it for me. If you are interested to know more, please look at the paper, which is available through open access at IEEE Explore. You can also stick around for the Q&A, or you can contact me at this email address. Thank you for your attention.
So thanks, David, for this very interesting talk and presentation of your nice technique. So far, I see a couple of people typing in Discord, so I will have the chance to start off with a question of my own. And that would be, so if I understand your technique correctly, you have these labels for the things you see on this on the next hierarchical level. So what happens if things are, so to say, hidden behind the model that you're showing? Do you also provide any navigation aids to find things that are hidden due to the camera angle?
Yeah, right. I mean, that's one of the problems that can happen in some models if you have a very unfortunate setup, like you mentioned, that let's say there is just one instance of a type of an object which is hidden somewhere. But we counter it with the specific sparsification technique, as we call it, because then you choose the instances to show, the representative instances, so that at least one of them is not, so that the representative that you choose is not hidden. So you can manipulate the cutting plane so that this cannot happen. The only way that can happen would be if the representative instances are obscuring themselves. So that's not really solved because we didn't encounter this problem with these models, but it could definitely happen in theory.
Okay, I see. Thank you. So we have a question from Laura Garrison. how do you handle the movement or possible occlusion of the labels as you orbit around the scene?
The problem of occlusion, so one thing is with the representative instances that I just described. The thing with resolving occlusions of the labels themselves, that's a problem that can be solved in theory. For this project, we didn't deal with it as much because it worked again reasonably well for most of the cases that we encountered. But there are ways how to resolve the occlusions of the labels themselves. I don't know if that exactly answers the questions, but that would be my answer.
Okay. I guess in the interest of time, we have to move on to the next presentation. So I still see a lot of clapping hands on Discord and YouTube. So thanks again for the nice presentation. And I would, of course, ask you and all the following speakers also to check the Discord channel afterwards to see if there are more questions popping up. Thank you. Moving on to the next presentation. This will be visual cohort comparison for spatial single-cell omics data. It's work done at Leiden University in Netherlands, and the talk will be given by Antonios Somarakis.
Hello everyone. Today I'm going to walk you through our work on the visual cohort comparison for spatial single-cell omics data. I will start my presentation explaining what the spatial single-cell omics data stands for. Spatial single-cell omics data are images depicting detailed tissue characteristics such as the abundance of many proteins or transcripts at subcellular resolution. Researchers have managed to identify the cellular borders and aggregate per cell their characteristics, enabling them to classify the cells into detailed cell types, forming complex and heterogeneous multicellular patterns. Current research findings demonstrate the clinical importance of these multicellular patterns, making imperative the systematic comparison of cohorts consisting of spatially resolved omics data with distinct clinical characteristics. And then the question arises: what is the differentiating factors among these two cancer images with distinct clinical cancer subtypes? Or more precise, which is the difference between the two cohorts of images with cancer type 1 and cancer type 2? Is it solely the amount of a dangerous cell type, or the coexistence of multiple different cell types in the same neighborhood? Are also the cohorts homogeneous? And finally, how can we validate that any findings are accurate and not a product of a cell segmentation or classification artifact? Traditional statistical approaches fall short on capturing the vast amount of possible cell type combinations and the plausibility introduced from the far from accurate cell segmentation and classification. Hence, to answer these questions, we developed a workflow for the visual comparison of two cohorts of segmented and classified tissue images with distinct clinical characteristics. Our workflow addresses the following tasks. First of all, we enable the user to compare the cohorts according to the abundance of different cell types. Most of the times, however, this is not adequate, and a detailed comparison based on the cell's microenvironment is needed. Also, throughout the process, we support the visual detection of outliers within each cohort and the validation of any findings, locating them on the tissue. To support the four identified tasks, we designed a multiple linked view system. The system is divided in three main blocks. The left block allows task 1, namely the comparison of cohorts based on the abundance of each cell type, and the right block, task 2, focused on the cohort comparison based on the cellular microenvironments. Task 4 relies on the location of any findings on both tasks. Hence, we show the corresponding images between the views. All three views allow task 3, the visual outlier detection. Let's examine now the rationale behind the design of each block, starting with a comparison based on cell type abundance. To do so, we calculate the amount of cells of a specific cell type for each sample and plot the samples in a one-dimensional scatter plot, according to their value. The more the lines on the right, the bigger its cell abundance is, as indicated from the highlighted cells on the bottom line. On top of the plot, we illustrate the density of the cohort distribution, forming a version of a rain cloud plot for both cohorts, using two complementary colors, orange and blue. To compare the cohorts, we superpose the two rain cloud plots. Blending the two complementary colors give us the light gray appearance, as it is illustrated in the overlapping area, emphasizing further their differences. To enable the comparison for all cell types, we use a small multiples approach, illustrating all cell types in a list, allowing the user to differentiate the cohorts, sorting them, or searching for a cell type according to user's prior knowledge. Here we search for all cancer cell types, placing first the plots which their cell types include the term cancer. In some cases, the classification of cell types is scattered in smaller subtypes, making cumbersome the differentiation of cohorts. To that end, we enable the user to aggregate their abundances, dragging progressively the rain cloud plots of user's choice into the drop area, where they're combined to the corresponding aggregated plot. Finally, selecting a line in the rain cloud plot, on the fly, the samples are shown in the tissue view and the cells of the corresponding row are highlighted, fading out all the other cells. At the end of this task, the user can identify the cell types that prevail in one of the cohorts, or cohorts with distinct clinical characteristics but same cellular abundances. In order to identify what actually causes this variability in the clinical functionality in the two cohorts, a comparison based on cellular microenvironment is needed. On average, each study has almost 30 different cell types, and each cell has 5 neighbors, forming its microenvironment. Hence, an exhaustive comparison of the cohorts based on all the different cell type combinations is impossible. To that end, we establish a two-step comparison, where first, for each cell of each cohort, we calculate how often occurs a cell of a specific cell type to have in its microenvironment one more cell type. Afterwards, we enable the user to compose combinations with specific cell types in the center and multiple cell types in the microenvironment, and compare the cohorts based on them. In the overview, we count the spatial occurrences following the same way we described before for each cohort, and then we subtract their values based on a user-defined metric, illustrating their explicit coding in a heatmap, where its x-axis represents the cell type of interest and the y-axis the cell types that exist in the microenvironment. For the heatmap, we utilized a divergent color map based on the previously described complementary colors. Having an overview of the spatial interactions, additional questions arise. Does the addition of yellow cells on top of purple cells in the microenvironment of green cells further differentiate the cohorts? Or is this specific spatial interaction combination present to all the samples of the orange cohort? To facilitate the user to answer these questions, we enabling him selecting a combination of interest, pre-populating the detailed view. Then, the user can start composing cell type combinations to further compare the cohorts. To illustrate the abundance of the cells in each sample that fulfill the selected combination, we utilize the same visual representation as we described before. Here, each line of the rain cloud plot shows the amount of green cells in each sample that have in the microenvironment purple cells. Below the rain cloud plot of the select combination, rain cloud plots are illustrated, showing the abundance of the cells that have in the microenvironment the selected cell types in addition to one of the remaining non-selected ones. To assist the user to find most differentiating factors, we added all the features of the small multiples we described before. So, reordering the plots places first the one that differentiates the most the two cohorts. In order to further explore whether a pattern, which includes an additional cell type in the microenvironment, further differentiate the cohorts, the user can drag the rain cloud plot and drop it in the microenvironment area. And then the user, following the same process, can explore in a data-driven way, including prior knowledge, all possible microenvironment combinations. As we did before, selecting a line in the rain cloud plot, we can locate on the tissue the featured microenvironment combination. In order to validate our design choices, we performed 3 different case studies. The case studies had varying characteristics, including the capturing modality, the amount of samples, and cell types, Here we will see how our workflow implemented in a standalone application is being used for a case study which features 20 tissue parts from a rare sarcoma case. The orange cohort consists of 13 images, indicating tumor areas with increased T-cell infiltration. We call this the hot cohort. The blue cohort, or cold cohort, consists of 7 images from tumor areas with little to no T-cell infiltration. The amount of cells in each image ranges from 2,678 to 23,334. Having identified that T-cells and macrophages differentiate the two cohorts, we move forward to explore whether their multicellular microenvironments can provide further clues on differentiating factors between and within the cohorts. Already from the differentiating heat map, we can see that B and CD4 T-cells in the B-cell microenvironment clearly differentiate the cohorts. By selecting the corresponding boxes in the heat map, we can see that these combinations are available in 8 samples of the hot cohorts, but they do not exist in the cold cohort, as indicated by the empty spatial view. The heat map selection also, on the fly, prepopulates the detailed microenvironment composition and enables the user to start exploring detailed microenvironments. Changing the x-axis of the rain cloud plot to absolute values, we can clearly detect 2 outliers. Clicking on them, they are highlighted in the tissue view. Here we can observe that the highlight area is part of an important biological structure, tertiary lymphoid structure. Afterwards, we reset the microenvironment composition and further compare the cohorts based on the cell type combinations that exist in the microenvironment of macrophages, illustrating with the yellow circle in the top row. The presence of CD4 T cells in the microenvironment offers a clear differentiation between the cohorts. Dragging it to the microenvironment area, we observe that CD4 and CD8 cells in macrophages microenvironment provide even better differentiation. Further differentiating the two cohorts and eliminating the outliers identified during the cell abundance comparison. Afterwards, we asked the case studies participant, in addition to another user of our system, to complete the questionnaire, including a standardized system usability test where an average score of 76.25 points was achieved, and open feedback questions where a participant mentioned that missed the functionality to simultaneously visualize all the raw high-dimensional images, and the sarcoma case participant who expressed his satisfaction discovering a tertiary lymphoid structure for which he was unaware before. Full details of this study can be found in our paper. Concluding with our interactive and data-driven workflow, we managed to compare 2 cohorts of spatially resolved omics data based on the abundance of the cell types existing in the cohorts, the cellular microenvironments they form, and the detection of outlier samples. In the future, we would like to enable the user to explore the microenvironments of 3-dimensional data and compare more cohorts. Finally, source code and binaries are available in our GitHub page together with a link for our archive preprint.
So, thank you, Antonios, for the talk and for presenting your interesting work. Again, first thing that comes up on Discord is, of course, a lot of applause. And so I wondered, so obviously the tool seems to be pretty useful for the experts. And so you got nice positive feedback. So I was wondering how well it would integrate in their daily routine. So how would it relate to other tools they've previously used to analyze this data, and could that be integrated easily?
Yeah. I think from the beginning of my PhD work, already 3.5 years ago, I'm working with this kind of data. Also, my previous work presenting on the Pacific VIS this year, we had a tool for the exploration and identification of, let's say, specific cell types. So actually, this work is a continue from my previous work and an extension, let's say, in order to enable the users that have already, with the previous tool, identify specific cell types to now compare them according to the cohorts. And as I have seen already, it's being used already in complementary to the previous tools. So I think it has been added well to their daily routine pipelines.
Pretty cool, yeah. Okay, thank you.
You're welcome.
Okay, let's see. I don't see any other questions currently. Thanks again for the presentation, and also please check the Discord channel for later questions.
Yes.
Okay. Now we will move on to the third talk in this session. This one will be given by Torin McDonald from the University of Utah, so it's a home game here. And the title of the talk is Improving the Usability of Virtual Reality Neuron Tracing with Topological Elements.
Hi, my name is Torin. In this talk, I'll be presenting our work improving the usability of virtual reality neuron tracing with topological elements. This work was performed at the Ski Institute at the University of Utah in conjunction with the Angelucci Lab at the University of Utah's Moran Eye Center. Our work focuses on an application in the neuroscience domain of connectomics, where researchers are working to reconstruct a map of the neural connections in the brain. This is done through taking high-resolution image stacks acquired through fluorescent microscopy imaging, as seen on the left, and then physically tracing out these neurons to produce a wiring diagram, like the one on the right. These image stacks need to be traced because the imaging process used to create them produces noise and gaps in the signal, necessitating people who have an understanding of neuron structures to decipher the data. Once these neurons are traced, the neuroscientists can then use the wiring diagram to gain a deeper understanding of fundamental brain functions and gain new insights about brain diseases and treatments. This process of tracing neurons falls into a larger neuron reconstruction workflow that first involves introducing a viral vector that induces fluorescence within neurons, rendering the tissue optically transparent, and then imaging this tissue with a confocal or 2-photon microscope. And as you can see, the tracing process is considered to be the bottleneck of this workflow. And this is because the image stacks that are produced are often complex and low resolution, and you can see, as you can see from the image on the left here. And there can be gaps in the signal or areas where multiple neurons come close together and can be difficult to distinguish them. The tracing process not only involves the time required to physically trace these neurons, but also decipher the data. Our work aims at reducing both the difficulty of neuron tracing and reducing the amount of time spent physically tracing neurons. So because of the time-consuming nature of neuron tracing, there has been a significant amount of research devoted to creating fully automatic reconstruction methods that vary from image processing techniques to machine learning models. However, in practice, these methods are not widely used because they struggle with the noise and gaps in signal from the data. And ultimately, manual intervention and post-processing is required in order to review these traces. And the step ends up being more time-consuming than manually tracing them. So as a result, there's a growing interest in using semi-automatic methods. A semi-automatic tracing method compared to manually tracing neurons aids the user by reducing the physical amount of tracing that needs to be done. This is accomplished by the user guiding an algorithm by roughly tracing along a neuron or providing start, end, and branch points. And what this allows is to increase the speed at which a user can trace a neuron while reducing the amount of time needed to review the resulting traces. So in the neuron tracing workflow, there's always this pervading problem where it can be difficult to determine the structure of neurons in noisy or ambiguous parts of the data. And in the 2017 VIS conference, Usher et al. Presented work that showed this problem could be alleviated using a virtual reality environment. And they found that working in VR makes interacting with the 3D data more intuitive because the users can visualize and interact with their data directly in 3D rather than having to scroll through a volume of image stacks. And this allows for a better understanding of the 3D structures within the data and leads to an increase in the speed and accuracy at which neurons can be traced. This system includes basic tracing features, including a paintbrush style of tracing, multiple rendering methods, and the ability to edit traces and haptic feedback for creating branching neuron structures. So we chose to build off of this framework to create a semi-automatic method to further alleviate the decision-making and fatigue problems associated with neuron tracing. So to do this, we wanted to create a method that quickly gives the user a visualization of the best possible path corresponding to the neuron they're tracing, and giving them a preview of what their trace would look like on demand, similar to what you see here. So to accomplish this, we need a graph-like representation of the possible neuron structures, similar to how streets and intersections are used in combination with shortest path algorithms to find directions. The Morse-Mail complex encodes the gradient behavior of scalar functions and intrinsically encodes the ridge and valley-like structures. In particular, we observed the saddle maximum arcs of the complex, shown here in red, follow the majority of the neuron structures. In our approach, we apply some image filtering and compute the one skeleton of the Morse-Mail complex as a pre-processing step, and then use it as the backbone graph structure behind our approach. I'll dive into this figure in a little bit more detail in a couple of slides, but it gives an overview of how the structure of our semi-automatic implementation leads to certain features that we developed. What we did to implement our semi-automatic algorithm is take the one skeleton of the Morse-Mail complex and we modified it slightly. There are instances where some of the arcs may merge, specifically the two saddle maximum arcs. So we insert a new node at each merger, and we call this modified one skeleton an MSC graph. And it maintains the property that any vertex along a path can be mapped to an arc in the data structure. What this allows is to create a fully connected graph representing every possible ridge-like path that then can be leveraged by a KD tree and shortest path algorithms to turn the neuron reconstruction task into efficient and interactive subset selection task on a sparse data structure. To make the algorithm more intuitive to the user who may not completely understand the underlying structure, we chose to hide the underlying structure and make it visible to the user as needed. We also follow common VR development practices where in order to reduce any visual fatigue or motion sickness, we keep any computation and rendering under 11 milliseconds per frame in order to maintain a frame rate of 90 frames per second. And when we developed and tested our initial prototype, we found that users spent a significant amount of time navigating the data and making decisions about where to trace neurons. So we designed our tool to not only increase the speed at which neurons can be traced, but also aid the user in this decision making. In the next slides, I'll go over some of the features we developed to accomplish these two goals. So the tracing algorithm utilizes a KD tree, which maps coordinates of the controller to the closest point on the MSC graph. This allows us to quickly query the MSC graph in order to display its local structure, and to give a visual to the user as to where their controller is relative to the MSC graph, we highlight the closest arc to the controller in orange. In the same vein, we also decided to create a flashlight feature, which gives a visual of the larger portion of the local MSC graph in green, and this enables the user to gain more insight into the data, especially low resolution or complex cases where multiple neurons may come close together. So to initiate a trace, the user clicks when they find a desired starting arc, which snaps the click to the closest point on that arc from the controller. And then as they hover around the data following a neuron, the shortest path along the MSC graph from their starting point to their controller is shown as a live preview in orange. Internally, we compute the shortest path on the MSC graph using the A-star algorithm, with edges weighted as a function of length and image intensity. And this live preview allows users to check in real time if the algorithm is producing the trace they want to commit. Once the user finds the desired endpoint, they click again and the trace is committed. This two-click functionality decreases the amount of physical user interaction that is necessary in manual tracing. And since a user no longer has to draw the exact center line of a neuron, we enable the user to zoom out to trace through much larger regions of the data faster. On the flip side, the user can also zoom in to navigate complex parts of the data. So for places where the algorithm fails or needs to be slightly corrected, we allow the user to easily switch between the MSE guided system and manual tracing. So to evaluate our method, we conducted a pilot study designed to compare our method to manually tracing neurons in VR. It consisted of 5 subjects, 2 experts, 2 undergraduates with prior experience, and 1 graduate with little tracing experience that spanned 2 sessions. Before the start of the sessions, users went through a training that consisted of a number of tracing tasks in order to learn the capability of both tools. The 2 sessions were completed at least 3 days apart in order to ensure that the user did not learn from previous sessions. And we recorded each section to extract all information about how users use the tool. Finally, after each session, we conducted a survey and had a free-form discussion to obtain qualitative feedback. So this table shows the average rate in voxels per second for each method across all 34 traces. We found that overall, on average, the semi-automatic method allowed users to trace faster than manually tracing, with a median speedup of 1.41 times faster. And in a real-world setting where traces can take days up to months, this is considered a significant improvement. This figure highlights the differences in the variability of trace quality produced by manually tracing versus the MSE-guided method. As you can see, when users trace with the manual method, they can easily drift off the center line of neurons, and the MSE-guided method eliminates this problem. In addition, through expert review and scoring metrics, we found that the overall quality of these traces were acceptable or even more accurate than those created manually. So one of our most significant findings was that the flashlight and trace preview features were found to be extremely helpful in the decision-making part of the tracing process, which we found consumed a lot of the user's time during the sessions. Users reported that in parts of the data that were low resolution or contained complex neuron structures, the flashlight and live preview features helped them in making decisions about what structures to follow, navigating gaps in the signal of the neurons, and determining branch endpoints. We got a lot of other positive qualitative feedback. Users found our tool to be less fatiguing and more comfortable to use for long periods of time. And what it really allowed them to do is focus on the data and decision-making rather than having to focus on making precise hand movements that are required for manual tracing. And as a result of the study, our collaborators lab is integrating our semi-automatic method into their workflow. Thanks for listening. I'm happy to answer any questions now, and if we run out of time, feel free to email me at torin@ski.utah.edu.
Okay, thanks for the talk. We already have questions on Discord. So Michael Sereno asks, can users add new nodes in case the MSC missed some?
So what we do is after we simplify the Morse mail complex and create the MSC graph, that is the kind of subset of arcs and nodes you're given. And in cases where that doesn't align with the neuron structure that the user is trying to trace, we allow them to quickly switch to the manual paintbrush mode. So this allows for quick corrections to the algorithm.
While we wait for more questions on the chat, I was wondering about the user experience of your tool. I think I've actually seen a demo of the previous version back from Will a couple of years ago. So I was wondering how you said that you took some measures to make it comfortable for users, so to maintain high frame rates and things like this.
Mm-hmm.
But do you have an estimate how long a user would be willing to be in the VR environment to do this? And did they like the using the controller, or would they rather have something like hand tracking, direct hand tracking? And so, yeah.
Yeah, for sure. So for the pilot study that we conducted, some of those sessions lasted up to 2 hours. And users reported not being fatigued after that amount of time. So while I wouldn't be able to give you a direct answer as to how long people would be willing to use the tool, we found that it wasn't fatiguing for the users. And then as far as the controllers and using controllers, we didn't compare that to using direct hand methods, but the motion is pretty simple. It just involves clicking a trigger a couple of times on the controller. Yeah.
So no errors or so due to the using the controllers. So people were able to use the controllers correctly. Okay. Good. We had one other question on Discord, but I have to admit I'm actually not sure if this was meant for you. So we had the question, how does your method take into consideration the 3D nature of the sample? But that might actually relate to the previous talk, I guess.
Yeah, well, to cover it, all the computation is done in 3D. So it's a 3D more small complex computation. And then in VR, you're immersed in the 3D
data. Okay. Maybe a last quick question. So having a volume rendering at 90 frames per second in 3D sounds rather impressive. So what machine do you need for this?
Yeah, so you can actually use just a normal desktop machine with not too expensive GPU. What we do is we only render a 256 cube subset of the data, which is streamed using an IDX format, which allows us to page in the data on demand. And so you could theoretically render any, like an arbitrarily sized volume for the tool, yeah.
Okay, all right. Well, thank you. With this, I would say, since I see no more questions, let's move on to the next presentation. So the next paper will be CMAT, Crowd Analytics for Medical Imaging Data. It's a joint collaboration, joint work by the Brookhaven National Lab, Memorial Sloan Kettering Cancer Center, and Stony Brook University. And the presentation will be given by Yuan Park.
Hello everyone, thank you for attending my talk. Today I will present a paper CMAT, Crowd Analytics for Medical Imaging Data. In this work I used two medical datasets, virtual colonoscopy and lung nodule. Virtual colonoscopy, called VIS, is a non-invasive screening procedure for colorectal cancer where a 3D colon is reconstructed from a CT scan of the patient abdomen. After the 3D colon is reconstructed, radiologists navigate inside the colon to detect a polyp, which looks like the left image. Similarly, in the context of lungs, radiologists have to interpret the 2D chest CT scans to look for lung nodules, the precursor of lung cancer, characterized by isolated spots not connected to the prevalent vascular structures. The right image showed an example of a lung nodule. Both the CT polyp and lung nodule detection tasks can be crowdsourced effectively, which can help reduce the interpretation time for the radiologist by letting them focus on the regions with high crowd consensus. The main purpose of our crowdsourcing annotation tools is that the crowd detect polyps and lung nodule candidates as good as radiologists, so it can be a crowd AD detection tools as a second reader to the expert radiologist. To achieve this purpose, we create the VC and long CT videos by navigating a column and rendering the videos of overlapping thin slab MIPS of CT slices respectively. We then present these videos to non-expert users or workers. Quality control is an essential component in the crowdsourcing application because of spammers. For example, randomly answering a task, and thus we added quality control object in each video to help detect spammers. Our goal is to improve crowd results by observing patterns and gathering insight into the crowdsourced medical data and ultimately help radiologists reduce their interpretation time. To achieve this goal, we provide a novel interactive visual analytics framework to visualize, classify, and filter crowdsourced clinical data, helping developers understand the crowd and improve their crowdsourcing framework. We also offer a set of visualization techniques to support exploring different aspects of crowd annotations. Lastly, we provide guidelines and lessons we learned from our framework. Let's look at our workflow. First, we collect the crowdsourced or workers annotation from our crowd annotation tool and ground truth annotation from radiologist. We then extract the workers logged event and annotated features and compute the detection rate of target object. Next, we incorporate several approaches to cluster workers annotations and their event. Lastly, we visualize all the data for the interactive explorations. In the next few slides, I will see the details of each step. To obtain crowd annotation for VC and long CT video, we use a web interface for both datasets on the Amazon Mechanical Turk platform, which is one of the most popular crowdsourcing platforms. Workers are provided with brief instructions including some example target object, police for VC videos, and long nodes for the long videos. The workers can play, pause, and rewind the video at will. Additionally, they can step to the previous or next frame. We also added the quality control object, 5 smileys for VC video, 1 gorilla for long videos in each video to help detect spammers. When the workers find the target or a quality control object, they first label it as a target or quality control object and then annotate the object by drawing a rectangle around it. We logged all the interaction events and store all the annotation information. We log the type of event that would occur as the workers perform the task and the timestamp of event. We also compute the detection rate of target object by comparing workers' annotations and ground truth annotations. Additionally, we calculated several workers and ground truth annotation features such as the number of missed quality control objects and annotated frames. After this processing, we clustered workers' annotation and workers' load event to aid the crowdsourcing application analyst in analyzing the annotations. Based on our preliminary work and previous crowdsourcing studies, discussion with radiologists, we characterized the following goals. Improve training cases based on exploring false positive of workers, explore missed polyps and see why workers miss them, explore the effect of missing quality control object on sensitivity and the type of worker, improve the current interface based on analyzing the worker's event log. Based on our goals, we designed the CMAT. This is the overview of our interface. Timeline view displays a summary of annotation for each videos. Frame view presents details of selected frames. Workers view shows the workers' annotation and corresponding event patterns. Class view displays characteristics of workers' classes based on event patterns. And matrix view shows a correlation between users' event patterns and their accuracy. In the timeline view, we reveal the overall annotation information for each video. This is an example of the timeline view. Let's take a closer look at the view. The left side shows a summary of annotation, and the right side displays the annotations. On the right side, stacked horizontal bar at the bottom of x-axis shows the ground truth target object, where the bar width indicates the number of frames in which the target is visible, and the bar color represents the target size. An analyst can select the specific frames in the timeline view, and the details of selected frames are then visualized in the frame view. Each line represents a target object. The x-axis indicates the video frame number, and the y-axis is the area of annotations. Representative annotations and images are illustrated by clustering workers' annotation within the same frame and across frames, respectively. We can display ground truth and a representative annotation of selected point on a line. We have two types of workers' view. The first one is the workers' view for each video, where we visualize the details of each worker's annotation per video. Left side displays a summary of workers' annotations such as number of identified target object, the size of object, and the missed target object by each worker. The right side shows the details of the workers' annotations and logged events. The second workers' view we provided is the workers' view for each worker, where I summarize all the tasks each worker completed. For each video worker annotated, we display the sensitivity of workers first low, the crowd consensus sensitivity second low, the number of quality control objects detected by the worker third low, and the worker's event class the last low. Our class view shows the distribution of 5 events for the 5 clustered event classes. The matrix view shows a correlation between event classes and the sensitivity of corresponding annotation for all videos. We present 2 case studies, VIS dataset and long-city dataset. The first study utilized the VIS data from 14 patients, yielding a total of 56 VIS videos. These 14 patients contains a total of 33 polyps. Each VIS video was viewed by 10 workers except for 2 which was viewed by 11 workers, and a total of 125 workers participated. In the second study, we used 15 videos. The video contains a total of 169 nodules. Each video was viewed by 10 workers, and a total of 30 workers participated. In the first video, the total number of frames annotated by workers was only 2.7% of the total number of frames in the video, yielding the potential to greatly reduce the required effort of the radiologist. In the long-city dataset, we achieved the wrong nodal detection sensitivity of over 90%. We also demonstrated our system to 2 analysts and a radiologist trained for VIS and long nodal detection. In general, the feedback for CMAD was positive. 2 experts especially liked our frame view because it shows what workers annotated or marked through an image with a bounding box. 2 experts found that the workers view for each video was interesting because it shows the details of workers' behavior patterns. Additionally, 2 experts mentioned that insight gained from workers' view from each worker was interesting because they could observe changes in the workers' behavior pattern even though they were still good workers. 1 expert also liked the matrix view because it provided him with a summary of workers. Lastly, all participants suggested that dynamic view configuration could be helpful since not all views are required for different types of analysis, even though each view provides complementary information. We have learned several lessons from our framework to help design future crowdsourcing application and visual analytics for medical crowdsourcing application. Some workers miss some quality control object but have good accuracy since they focus on the target object. Thus, we cannot ignore workers who miss a few quality control objects. Additional training and more examples of tricky polyps long nodules might help improve the ability of the crowd in identifying polyps which looks like a fault. Since some workers may perform well consistently, we can rank workers and consider the expertise level of the worker when compiling workers' answer. For VC datasets, some polyps are only visible from certain fly-through. We can register different fly-throughs and visualize corresponding parts in each video. To sum up, we presented CMAT, a novel visual analytics system for the interactive exploration of medical data annotations. Our designed framework provides various insight into crowdsourced clinical data which cannot be provided by other tools. Our case study demonstrates the usefulness and effectiveness of our framework. Thus, CMAT can help crowdsourcing application analysts or developers to design better crowdsourcing study. In future, the result of our framework can be integrated into clinical workflow as a second reader or annotated data for deep learning. Thank you.
Okay, thank you for the interesting talk. I already see people applauding again on the chat. And while we are waiting for questions, I was wondering about the last thing you also mentioned on the future work. So this seems to be an ideal tool to create, so to say, a training data set for a machine learning, a neural network based approach, right?
Yes.
So given that you still don't have, probably don't have a huge number of data sets, but could you compensate this then with the number of people doing the study and then providing annotated data?
Yes. So what we are trying to do with this framework in the future is that we just ask the small amount of data for the expert users and get the annotation, and then in the meanwhile we get also data from the cloud and then integrate those data while we training the models.
Okay, thank you. I actually don't see any further questions right now in Discord. There will probably some pop up later on. Since you're a bit behind schedule, I would again ask you to also check Discord later on. And thanks again for the nice talk. Very interesting work. And yeah, then we would already move on to the last talk of this session. This one will be given by Julian Kreiser from Ulm University here in Germany, or at least where I am. And the title of this paper is Void Space Surfaces to Convey Depth in Vessel Visualization.
Hello everyone, and welcome to my talk. My name is Julian and I'm from Ulm University in Germany, and there I'm working in Timo Ropinski's visual computing group. And today I'm going to present our work, void space surfaces to convey depth in vessel visualizations. Most current vessel visualizations convey functional parameters like wall shear stress and pressure in hemodynamics, elasticity, the occurrence of plaque or vascular constrictions directly on the geometry of interest. However, this makes it difficult to visualize additional depth cues for complex or thin structures at the same time on the same geometry, and usually requires some sort of blending or overlay. This has the disadvantage that such techniques add up visual clutter, which might result in cognitive overload or overdraw of important information. The image you can see here is an example from the work of Behrendt et al. They combine a parameter mapping with a pseudo-chroma depth color map on the edges of the surfaces to enhance the depth perception. To overcome the limitation of displaying depth cues together with additional information on the same geometry, like in the image on the left side here, we take advantage of the empty space, the so-called void space in our work in between the vessels, shown here on the right side. We generate a height field, which then serves as a canvas to convey depth without having to interfere with other visual encodings on the vascular structures. To be clear about the rest of the talk, this concept is what I will refer to as void space surfaces. Let's start with a quick example. If I would ask you which of those two branches is in front of the other, your answer would probably involve some guesswork or tracing the vessels back and forth to get a clearer understanding of the scene. However, when we put our void space surface in the background of the image, you can clearly see that the right branch comes more out of the screen and lies in front of the left one. Here is another illustration on how to interpret void space surfaces extended into 3D space. This video clip shows the depth differences of the vascular structure in a transition from the normal user's view in 2D to a side view in 3D and back. Here, with an side-to-side comparison of the same scene as the single frame, as you have seen from the video clip before. Let me now explain how we generate the actual surface in the background of the geometry. To get the void space surfaces, we interpolate the depth values around the silhouette of the geometry, like here in black. In this example, the colored patches represent closed regions in screen space, which are processed independently. And from left to right, there would be 1, 2, and 6 different sections, depending on the zoom level. We have chosen to use an inverse distance weighted interpolation with a power parameter to control the details along the silhouette. Here you can see the current pixel in blue, for which the depth is calculated, and the pixels along the silhouette are marked in orange. We now calculate a weighted sum for the depth at each silhouette pixel xi with the inverse distance to the point of interest x0, and raise that distance to a power parameter to control the smoothness of the interpolation. Lastly, the weighted sum is normalized by all weights. This row of images show how a variation of the power parameter p influences the smoothness of the interpolation. P equals 1 corresponds to a linear interpolation, and p towards infinity would result in a nearest neighbor interpolation. So how does that translate to actual vessel geometry? This picture shows how a 2D side view would look like with two vessels connected by an interpolated surface. The vessel structures are connected at their widest point from the view direction, as those would correspond to the silhouette in screen space. The black outlines are the visible parts of the geometry to the user, and the orange line represents the void space surface. Now we can treat this generated depth map as a height field and apply shading to it, for example. This anchors the vascular geometry onto the surface and improves the depth perception at the same time, as you can see here in the right image. Here's another example of two images which have been generated with a different power parameter. On the left, the inverse squared distance is used, and on the right, a power parameter of 5 to amplify details. Additional to shading, I'm going to present other enhancement strategies we employ to further aid the depth perception using various side field visualization techniques, which have proven to aid depth and shape perception. And we found color coding and isolines particularly helpful when it comes to vessel visualizations. In the first two images, you can see a chroma depth and pseudo-chroma depth color mapping to create the illusion of depth in 2D images. Additional isolines help to convey local changes, and especially at small depth differences. I should note that these enhancement techniques are not all which could potentially be used, but rather a subset of what void space surfaces are enabling. Isolines can also be used to quantitatively display depth differences on the surface. Here you can see isolines with a uniform line width on the left and a mix of primary and secondary isolines on the right, similarly to the markings on a ruler. Having secondary isolines helps to reduce clutter in the image, to leave more room for other information to be displayed as well. As you can see in this example, even with a full chroma depth color coding with additional isolines on the left, shading is a very strong indicator to use for shape and depth perception. The appearance of ridges and valleys clearly help to communicate which structures are in front of another. With the void space surface now in place, the geometry of the vascular structure can now completely be used to map additional functional parameters, like blood pressure or wall shear stress. Here are some examples with different color coding in the background, and on a geometry to separate the depth cues from the map parameters of the vessel structures. To now evaluate the perceptual performance of void space surfaces, we conducted 2 quantitative lab studies to compare against other established vessel visualization techniques. And in particularly, we were interested in how fast and accurate participants can judge depth. Our first study focuses on the comparison of the underlying depth cues itself. Void space surfaces is compared here to the direct colorization of the vessels and a neutral baseline, all without functional parameters. We use the chroma depth and pseudo-chroma depth color schemes for the void space surfaces, like here on the bottom right, and directly on the vascular structures as seen on the bottom left. Then we asked the participants to judge which of the 2 marked structures in the image appears closer to them. The structures were marked with a crosshair, like you can see on the top left, which is also present in all other images as well. For the results of this first study, all of the depth-enhanced visualizations performed similar and outperformed the neutral baseline rendering in terms of accuracy. The left graph shows this with CD as the chroma depth and PCD as the pseudo-chroma depth versions. As to response times, we found that the methods which use depth cues directly on the vascular surface are faster than all 3 other configurations. The second study compares our best performing setup, which was the pseudo-chroma depth version, to the method from Behrendt et al. Here on the bottom image, and again with a neutral baseline. All with added functional parameters, this time now on the vascular surface. And we have chosen to involve experts from the fields of medicine and biology, which are usually more familiar with such datasets. On this study, we found significant differences between the compared visualizations only for accuracy. The response time was similar for all 3 techniques. Void space surfaces performed more accurate than the state-of-the-art from Behrendt et al., which itself, and to our surprise, had no significant difference to the neutral baseline. We believe this originates from the thinner parts of the vessel structures we have used in our tests compared to the geometry in their study. The pseudo-chroma depth colorization of the vessel's edges in such a case is not prominent enough, usually. This was also confirmed by most participants. They mentioned that they could often just distinguish between red and blue, barely differentiate similar colors on the gradient. At this point, I should mention that void space surfaces also have limitations. The most obvious one would be the necessity of actual void space between the geometry of interest. However, vascular structures usually provide such gaps quite nicely, especially with increasing scanner resolution, but it still has to be considered. The other limitation we could identify is if an object shares no contour with the background and is therefore not attached to the void space surface itself. This happens when a structure lies completely in front of the other one and gets occluded from the background side, as this illustration here shows. In this case, only occlusion and shading cues support the user. To quickly summarize, void space surfaces are a technique to move depth queues away from the geometry of interest. This allows for an exclusive display of functional parameters on the vascular surface without disturbance. Additionally, this technique can also be implemented for real-time applications, which we already also did. All right, that would be everything from me today. Thanks for joining. Hope you had a good time.
Okay, thank you, Julia, for the presentation. We already have the first question on Discord. And this is, do you need to regenerate the void space surface on view rotation? So if vessels occlude each other, and how fast is that?
Yes, we do have to recalculate that because the technique works in screen space, and we do that via compute shaders. That works in real time. Of course, I think we tested on full HD images. For that, it worked without like even super optimizing it. But of course, if you like scale up the image because it's a screen space technique, then it gets heavier. But for regular screen sizes like full HD, that's not a problem.
Mm-hmm. Okay, thank you. Tobias Isenberg asks, what happens when the void space is needed for something else? For example, for other autonomy, can some semi-transparent setup be used maybe?
We haven't tried transparency since we didn't have other geometry in the scene. But I think as long as let's say the chroma depth color mapping is still visible enough to the user, I think it still can support it. Otherwise, what you could also do is just use like secondary geometry, like a tumor, for example, into the scene and also generate around the void space itself in these regions.
Mm-hmm. So Cameron asks, well, first, thanks you for sharing this excellent work, and says, I have a small question. Why mention the sex of the participants of your user study if it's irrelevant? Or was it relevant in some way? So did you see any differences for Leo?
I think, or not, I think we included them because usually there's a differences between males and females in terms of, let's say, like color perception or depth perception, because men usually suffer more often from, let's say, yeah, like what is called like red-green blindness or things like that. And I think that's even like 10 times more common in males than in females. So we just recorded that for like the completeness sake. But usually if you don't have any visual impairments, there should be no differences between males and females.
Okay, thank you. So maybe a quick question for me. I wondered, did you think about comparing this method to stereoscopic display? So for example, a virtual reality setting? Do you have an idea how it would compare?
I think in VR, it's not really, let's say, fair because you have like 3D vision out of the box in VR anyways.
Mm-hmm.
So we focused on, let's say, like a pure 2D display, let's say like a surgeon has in the operating room on a display, while simultaneously using tools to operate on the patient so that he has like the, let's see, static 2D images on the screen while operating. Or if you print out stuff, then you only also have a 2D representation. That was the main use cases we intended it for.
Sure. Oh, of course, it would not be a fair comparison. That's of course clear. The question is just how much worse would it be? Maybe it's even on par with stereoscopic view. So I guess that could be interesting.
Yeah, that could be like a topic for future research for sure.
Okay. I think we have time for one last quick question from Pavel sorry for messing up. How effective is this method when we look at large sections with maybe tens thousands of vessels? Have you looked at that?
No, unfortunately not. We didn't have like such huge data sets. But as long as there's enough void space in between vessels, and it's not only, let's say, a pixel in between vessels, then of course the technique is not effective. But if you have like increased scan resolutions, like in the future, and you have enough void space in between, I don't see a huge issue if it's not only like a very small part of the image.
Okay.
But we haven't tried, so.
Okay. And Pablo comments that he has such data sets, so he would love to chat offline with you. So I would say this concludes the session. Thanks to all our 5 speakers for the wonderful presentations again. As I said, I think you already saw a lot of clapping emojis on Discord. I very much enjoyed all the presentations. And again, I would ask all the speakers and maybe the co-authors to check the Discord channel during the next days to see if other questions pop up. So yeah, thank you. And of course, thanks everyone for coming.
How to know if a graph is correctly drawn? Are all the aesthetic properties preserved? To answer these questions, we currently use quantitative methods that require graph structure and are computationally expensive. We propose a new method to directly compute these metrics from graph layout images using convolutional neural networks. Below are the results. To know more about our approach, please attend our talk.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. With Tableau, you can keep on asking questions in the data until you discover the root cause. Share your analysis securely. Your entire organization can access these interactive dashboards from any browser or mobile device to find their own answers. Tableau. Answer questions at the speed of thought.
We used data from today's United States to recreate 7 abolitionist data visualizations, which were originally presented by W.E.B. Du Bois in 1900. Comparing the modern and historical versions, we can see continued racial disparity in America's geography, economy, education, and justice systems. This view of the past alongside the present invites us to look at the historical context for modern-day abolitionist movements.
A short overview of the presentation for a simple pipeline for coherent grid maps. In a grid map, we transform an input map to a tile arrangement. However, current approaches do not perform well on complex maps such as the Netherlands. We propose a pipeline that generates high quality and coherent grid maps even for complex data using a simple pipeline.
We present an exploration into co-located collaborative immersive analytics in a prototype called Fiesta, the free-roaming immersive environment to support team-based analysis. The prototype allows groups of users to visualize and explore their data in a room-scale environment, which we used in an exploratory study involving 10 groups of 3 participants. We focus on the roles which 2D surfaces have, as well as how users leverage the 3D space during both the individual and collaborative work.
The make of continuous color maps. We present the CCC tool, a new web tool for the creation, editing and analyzing of application-specific color maps. For the precise definition of color maps, we developed a new color map specification. This new structure increases the editability by reducing the numbers of colors to a minimum.
Hi, please come see our talk if you are curious about how to encode your data so that it can be efficiently queried later at different fidelity levels by fetching only the relevant bits from the disk. The talk is titled Efficient and Flexible Hierarchical Data Layouts for a Unified Encoding of Scalar Field Precision and Resolution.
As visualizations begin to use color, shape, line, and form in new ways, legends often go overlooked as essential to interpretation. To address this issue, we created a web-based interface that renders and formats legend entries, presenting the dynamic properties of glyphs, lines, and surfaces, and increasing nuanced object recognition in artist-driven 3D scientific visualizations. Each legend entry includes the variable name, full value range, and artist-chosen artifact.
This paper explores the efficacy of using narratives as a medium of explaining the causality in causal networks. Now, towards this end, we provide you a glimpse of CausWorks, a causality visualization system for generating narratives given a set of interventions and objectives for a causal model. And we further validate CausWorks through interviews with experts. Please read our paper to know more. Thank you.
To uncover football tactics, we create spaces and times. We use time query, construct spaces, construct times. Our approach was used for reconstructing team tactics in different classes of game situation. Our domain experts are happy, and the Germans shall win again and again.
This study proposes a taxonomy of sentiment data visualization based on visual metaphors. In order to create a taxonomy, we enhanced a group of classification criteria. And then, we classified a total of 35 papers published in the last 20 years. As a result, we presented the guidelines for researchers doing research on the sentiment visualization and visual metaphors.
Our paper describes the motivation, ideation, and prototyping of a novel visualization tool to support voice training for transgender people. We centered our design around user needs and lived experiences in order to support self-determination of gender expression. In so doing, we made novel inroads in how to visualize speech data and opened possibilities for future work in the design of visualizations that prioritize user agency.
Dimensionality reduction is an important tool for the visual exploration of complex high-dimensional data. Most existing techniques focus on preserving the intrinsic geometry of the data during the projection. However, important information can be encoded within the global structure of the data. To address this, we present TopoMap, a dimensionality reduction method that provably preserves the topological features of the data during the projection.
How do people really make use of space to spatially organize their ideas? To answer that, we collected 280 images from the web, which came up by searching keywords such as brainstorming. And we found spatial organization patterns and interesting observations about how people make use of space.
In this work, we present an atmospheric model tailored for the interactive visualization of planetary surfaces. Our model provides a complete framework for the absorption of light by small and large particles, such as the oxygen and ozone molecules and Mars dust particles. We demonstrate our results using Earth and Mars as examples.
Nowadays, technology is developed for a single audience, assuming that one size fits all. Nevertheless, we all have individual differences, such as personality. Taking into account how personality affects preferences, we study whether it has an effect on hierarchy, evolution over time, and comparison context regarding information visualization. We leverage all personality variables from the 5-factor model and the locus of control with correlation and clustering approaches, showing promising results.
This study presents a method to explore hierarchical data by improving ROD-VIS, one of the visualization techniques for multidimensional data. The user can set options for nodes and check the data in groups or individually in the ROD-VIS. 3 subviews at the right enable the hierarchical clustering structures of selected data and allow the user to grasp the features of selected data. As a result, this system allows more detailed insight into the data while solving the node overlapping problem.
Existing visualization tools for deep learning classification are mostly working on natural images with mutually exclusive classes. The classification of X-ray scattering images is more challenging since their multiple structural attributes have complex relationships. So we built an effective visualization system to study the model performance and to find questionable labels or outlier images for scientists' further improvement.
Designed in response to the COVID-19 pandemic, the novel visualization technique we are calling GOWNS accommodates continuously incoming and increasing case data without distorting past data. Layering linear scale graphs shows both small and large numbers. This allows more detail to be seen when scrubbing through time and comparing regions by realigning the GOWNS based on case numbers.
Data GIFs are enjoying increasing popularity on social media as a format for data-driven storytelling with visualization. Simple visual messages are embedded in the short animations and are played in automatic repetition. Here we present a structured design space and a set of design instructions to create effective data GIFs.
We propose V2V, a comprehensive pipeline for variable selection and translation for multivariate time-varying data. Our approach contains feature learning, translation graph construction, and variable selection. To demonstrate the effectiveness of our approach, we compare histogram matching, pixel to pixel, and cyclogram to evaluate variable translation. In addition, we also compare an information-aware framework to evaluate variable selection.
What is insight? It is a question that has bothered visualization researchers for a long time. We're wondering whether visualization practitioners think about data insights in the same way as visualization researchers do. So we interviewed 23 visualization practitioners to understand how they think. To find out more about what we found and the implications to the visualization community, take a look at our paper.
We propose a method palettaler that fully automates the coloring process of categorical data and produces discriminable results. Based on both color and data spaces, our method incorporates 3 scoring functions into a customized simulated annealing algorithm. We apply our method to different types of visualizations and compare it with the state-of-the-art palettes through a controlled user study.
Choroplast maps are popular. However, we have better encodings for numeric values. Check out the prism map. Worry about occlusion and perspective distortion? Let's go immersive. Want to have both of them? We present tilt map, a novel interaction technique for transitioning from a choroplast map to a prism map to a bar chart to overcome the limitations of each. Believe me, tilt map opens up many research possibilities.
Virtualizations don't have to be static. In fact, sometimes it is the visualization that is static, but the viewer is moving. And sometimes both of them move. We present a first design space of visualizations in motion, an implementation of this design space, and we are working on a user study.
We propose a localized approach to topological simplification called LTS, which computes the exact same result as the current state-of-the-art simplification procedure implemented in the topology toolkit. But LTS is an order of magnitude faster as it utilizes shared memory parallelism and constrains computationally expensive procedures onto small subsets of the domain. This approach significantly accelerates an essential part of topological data analysis pipelines.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau. Answer questions at the speed of thought.
Capturing analytic provenance is important for refining sense-making analysis. However, understanding this provenance can be difficult in distributed collaborations. We present a novel concept, crowd auditing, as a way to help debug distributed sense-making. We implemented the concept in a system called CrowdTrace that visualizes and traces the analysis provenance and elicits feedback to improve the analysis.
Hi, everyone. It is a work about graph visualization. With one node link diagram, dragging node can change the layout of the diagram as you like. But how about more diagrams? Dragging nodes can be laborious. Please come to our talk about exemplar-based layout fine-tuning for node link diagrams. We will give you our ideas.
Functional safety analysis is an automotive systems engineering technique that helps minimize unreasonable risks associated with automotive electronic and electrical systems. We developed a visual analysis tool, Safety Lens, to assist domain experts to perform exploratory analysis of a single project as well as comparative analysis among multiple projects.
The search for high prevalence of crimes ends up neglecting sites where certain types of crimes are frequent but not sufficiently intense to be considered statistically significant. In this paper, we proposed a technique centered on NMF to identify hotspots based not only on the intensity but also on the frequency. We introduced a visual analytic tool to support the domain expert in their analysis, whose usefulness of the system are highlighted by comparisons and case studies with real data.
Large displays are beneficial for visualizing vast amounts of data. However, there are challenges regarding perception, personalized views, and managing data complexity. To address these issues, we propose to combine large interactive displays with personal augmented reality. We present a design space, as well as several visualization techniques, such as curving the whole screen, embedded visualizations, and many more.
Usual collaborative systems propose a public and private environments, the latest possessing visibility rights. Subjective views are spatial objects perceived differently per user. However, a break in the social space may happen during exploration. Our visualization context strives to link efficiently multiple related visualizations together around a common object. We discussed about the designs of a stacking layout, virtual linking layout, and a mix between them.
In this study, we designed PassVisor to fully analyze the passing dynamics. Users can hover on a pattern diagram to see all the passing patterns, its characteristics, and how these patterns distributed over phases. They can further click on a pattern to see its phases, select one phase to see the passing, and inspect the prior statistics during passing.
Hi, everyone. It is a work about graph visualization. With one node link diagram, dragging node can change the layout of the diagram as you like. But how about more diagrams? Dragging nodes can be laborious. Please come to our talk about exemplar-based layout fine-tuning for node link diagrams. We will give you our ideas.
University directories routinely become outdated and often lack the information necessary for understanding a researcher's interests and past work. To assist in the exploration of research talent, we have designed PeopleMap, an interactive tool that maps out researchers using NLP techniques. By using PeopleMap, users can explore the diversity of research at an institution and identify potential research talents.
Studies in HCI follow similar steps in explaining hypotheses, laying out a study setup, and reporting results. Many of these decisions are buried in dense scientific text. We contribute a set of examples of how data comics support that reporting, providing an initial set of concrete ideas for how to communicate common stages of HCI studies and statistical analysis.
Generating a stacking ensemble of models is usually a complicated trial and error process. In order to deliver the best and most diverse stacks to the user, we propose StacksNVis, a visual analytics system that supports an iterative process. With different algorithms and quality metrics, instance and feature selection, model optimization, and more.
We are introducing Polyform, an interactive tool for analyzing the intergalactic gas and dark matter filaments, together known as the cosmic web. Here we see Polyform interactively reconstructing and visualizing the cosmic web estimate over 37,000 galaxies from the Sloan Digital Sky Survey dataset. Polyform uses the Monte Carlo Fissure machine algorithm inspired by the foraging behavior of the Physarum polycephalum organism, also known as slime mold.
Classroom participation data disaggregated by race and gender can enable teachers to reflect on their implicit biases and consciously enforce participatory equity in their classrooms. In this work, we explore a design space for representing classroom data to enable teachers to compare contributions across students and student groups, as well as compare participation rates of the different student groups with their demographic compositions.
To generate a visual data story, one needs to learn a lot of techniques such as data analysis, visualization, and scripting. To lower the technique barrier, we introduce an intelligent system, Calliope, to generate visual data stories directly from an input spreadsheet in a fully automatic process. Using Calliope, a good visual data story is just a click away from us.
When doing the cell division for an embryo, plant biologists use several tools to get segmented 3D dataset and build the hierarchy tree manually by traversing and analyzing neighboring cells and deciding on which neighbor is the sister based on their knowledge. In this poster, we present an interactive system for biologists to help with analyzing the development of plant embryo.
Over hundreds of tools exist for visualizing genomics data, leaving the analysts with a large design space to analyze before finalizing the visualization. We present a data and task-based recommendation system. The recommendation system uses a sequential model to suggest appropriate visualizations to users. To learn more about our recommendation methodology and results, please visit our poster.
Atmospheric convection is one of the most important phenomena in meteorology. It is responsible for cloud formation and storms. Yet, the measurements of the convective processes are expensive and scarce. In IsoTrotter, we collaborate with a domain expert to build an empirical model of the atmospheric convection based on paragliding data. We design a novel visual parameter space analysis technique using isostructures in the parameter space to explain and validate the model.
Rendering large volumetric datasets is still a challenge for real-time applications. Wouldn't it be nice if you could just render the dataset at a low screen resolution and your neural network upscales it to the full resolution again? We propose a network architecture that does exactly that and can outperform a full resolution rendering for large datasets. For quantitative and qualitative evaluations, come to our talk.
urban motion, illustrating the movement of population from billions of sparse trajectory records. Chang'an Avenue correlates with movement flow. The regions with the largest flow volume. Zoom into the region. The southern Huilongguan region is mostly in blue, cut by G6, which could account for its commuting issue. Rainstorm in Beijing, movement speed is much higher on average. At the remote regions of Beijing, there are few local movements.
DataBreeze is a multimodal visualization system that lets people interact with unit visualizations using pen, touch, and speech. By supporting multimodal interaction, allowing users to manipulate, refine, and customize the view, DataBreeze enables a novel, fluid, and flexible data exploration experience, enabling a seamless transition between global and local level data exploration, spanning the entire dataset or a subset of the data.
Uncovering causal relations in event sequences can benefit decision-making in various applications. We present an interactive visual analytic system, Kouřil. It incorporates an algorithm with a user feedback mechanism for learning causality in event sequences under the supervision of human experts. It also provides a set of visualizations to support causal exploration, verification, and comparison.
Tableau helps you see the stories in your data. It's designed to help you be smarter, so you can make better decisions faster. Connect to the data you care about. Sort, highlight, drill down, or filter your data in seconds. Add calculations to extend your data. With Tableau, you can keep on asking questions in the data until you discover the root cause. Tableau, answer questions at the speed of thought.
Machine learning advanced the state-of-the-art in many fields. However, for VIS, it has just begun recently, partially due to the lack of training data. To support data-driven research in VIS, we simulated and released a 16 terabyte dataset containing 8,000 unstudied 2D vector fields. We demonstrate its usefulness in a Lagrangian transport upsampling problem by employing CNNs on different simulated and measured datasets.
Mode surfaces are the generalization of degenerate curves and neutral surfaces. In this paper, we provide novel analysis on the topological structures of mode surfaces. This allows us to not only better understand the structures in mode surfaces and their interactions with degenerate curves and neutral surfaces, but also develop an efficient algorithm to seamlessly extract mode surfaces. We apply our analysis and visualization to a number of solid mechanics datasets.
Typically, black hole visualization takes a lot of compute time, but we achieve a quite faithful reproduction at interactive rates on a standard computer. For this, we make use of a novel adaptive grid approach to focus calculations where needed. Coupled with filtering and interpolation methods, we can obtain high-quality imagery. Our approach accepts star catalogs and environment maps and generates the resulting deformations in real time.
Table cartograms are a recent form of data visualization that embed tabular data in a grid of quadrilaterals, like a heat map that has been areaed rather than colored. There are lots of different valid outputs for a single input table. Unfortunately, algorithms found in prior work prevent exploration of this design space. We present a web-ready algorithm for creating table cartograms using an optimization-based approach that supports a wide variety of outputs and aesthetics. Find out more at this address or come by our poster.
We present an investigation into design judgment in DataVis practice. Our findings show how different types of judgments influence design decision-making. Our findings also show that judgments occur in complex and layered ways while designers move through their process. We discuss implications for future research and pedagogy.
Ensuring the usability of a dataset is often tied to data wrangling. We can use provenance to log the employed steps, but these alone will not give us meaningful insights if data